Okay, so up to now we've talked about the organization of DNA in our cells, the fact that it's actually DNA and protein, and it's this mass called chromatin. And when our cells are gonna divide, it coils up into these individual chromosomes, and we've talked about the structure of a single chromosome. So now we're going to talk a little bit about how those chromosomes are organized. So first of all, our body cells, also called somatic cells, it means body cell. In each of our body cells, we have two sets of chromosomes, meaning we have two copies of every chromosome. And there's a name for this. This is called our diploid number, 2n. And the reason why we have two copies is because we got one set of chromosomes from our mom, one set from our dad. When sperm and egg unite, you now have two copies of each chromosome. And I'll show you a diagram on the next page that will help to illustrate this. Now, each species has its own diploid number. Our diploid number happens to be 46. So that means that in every body cell, there's 46 chromosomes. And again, that's called 2n because there's two copies of each one. Sperm and egg, what happens is our chromosome number gets cut and we only give one random copy from each pair to the offspring, and that would be our N number, which we'll talk about later. And our N number is 23, meaning there's 23 base chromosomes in a sex cell, a gamete, sperm or egg. But since our body cells came from the fusion of a sperm and an egg, they basically have two copies of every chromosome. Fruit flies, for example, have, I think, eight chromosomes as their diploid number. Certain species of corn have like 300, so there's not necessarily any rhyme or reason that the more you have, the more complicated you are, or anything like that. So this may help to clarify. This is called a karyotype. This is a picture of the chromosomes that's generated. You could make one of these or have one of these made. They would take a blood sample and treat it with certain chemicals and use white blood cells actually, and then they would get your chromosomes, and then by computer they would sort them out and pair them up. And so you have, this is your 23 pairs of chromosomes, and you see that they're lined up. You have two copies of chromosome 1, two copies of chromosome 2, and for example, the two copies of chromosome 1 carry the same genes. So for example, let's just say chromosome 1 controlled blood type. That would mean that you would have a gene for blood type on one of the chromosomes, and you'd have a second gene for blood type on the other one. It doesn't mean that you have that the genes say the same thing. If blood type's carried on chromosome 1, then that would mean every person in this room would have two different copies of the gene for blood type. Now, if they happen to be a copy of A and a copy of B, then your blood type would be AB. If it was a copy of A and another copy of A, your blood type would be A, etc. So the actual DNA may code for a different form of a gene, but if, like I said, if eye color or hair color or blood type or whatever is carried on chromosome 1, they can always look at chromosome 1 to see that, and that's how they've been able to figure out. For example, the location of certain breast cancer genes or certain diseases. They can literally go in and they can say, well, I know this disease is carried on chromosome 4. So let's look at both copies of chromosome 4, see if that DNA sequence for that particular disease or whatever is there. If it is, then they would know that you had that disease. But they would know that they only have to look at chromosome 4 because that's the chromosome pair that would have that gene. You couldn't tell that by looking at this, by the way. The only things that a karyotype would tell you specifically why somebody would have one done is it would tell you if there was a mutation in the number of chromosomes. For example, an extra chromosome right here would be what they would look for Down syndrome, an extra chromosome 21. You could also tell from this karyotype this happens to be a male because the last pair are called sex chromosomes, and this one has XY. The Y doesn't really look like a Y, but it's a very, very small chromosome, versus a female would have two copies of the X. So we'll actually talk a lot more about chromosomes in the upcoming chapters, but that just gives you sort of a preview. And this is another diagram, basically showing the same thing. This is they're X shapes, but the two chromatids sort of stick to each other. So these are also dyed. You can actually see little bands in these, but it's the same idea. This would also be a male. There's an XY there, and I don't think there's any kind of defect in this particular one. So, okay, so that brings us to the cell cycle. And the cell cycle is exactly what it sounds like. It's the life cycle of a cell. A majority of the time, the cell is in what's called interphase, meaning in between mitosis. It's the time when the cell's not dividing. And it's the most important time in the life of the cell because that's when the cell is doing whatever its job is supposed to be. So if it's a muscle cell, it's involved in making muscle proteins, contraction, etc. If it's a white blood cell, it's involved in phagocytosis, fighting diseases, whatever it may be. So that's called interphase. And interphase is subdivided into 3 separate parts. And originally, when they first discovered the cell cycle, it looked like there was this big portion of time where nothing was happening, and then there was some stuff happening, and then a big portion again. So originally, the G1 and G2 stood for gap phase 1 and gap phase 2, this big gap of time where nothing seemed to be happening. Thing is, they just couldn't see what was happening because since the chromosomes in the nucleus are very big and easy to see, it was easy to see when a cell was going through synthesis, and it was easy to see when a cell was going through mitosis because that's when the chromosomes were all coiled up and it was easy to see stuff going on. They didn't realize that during G1 and G2, this is actually when the cell was doing its most important jobs. So now, when you think of G, don't think of gap, but G1 we say is primary growth. So what's going to happen here, this would be a cell that's just been formed out of, let's just say, out of a previous mitosis, and the main thing that's going to happen here is the cell is going to grow. One thing that's going to need to happen is if the cell just divided, that means it's going to have half of the endoplasmic reticulum and Golgi bodies and all those things that it had originally. So this would be a time when the organelles would probably divide, the cell would rebuild, make new cell membrane, make new membrane proteins, the mitochondria would divide, etc. So that's primary growth, and we'll talk a little later that if a cell is never ever going to divide again, then it actually will go into what's called G0. And it will actually just stay in G0 because it's never going to need to divide again. But a normal cell, if it is going to divide again, will go from G1 to a stage called synthesis. S is for synthesis. And this is where the chromosomes make copies of themselves. If you recall, we talked about how a chromosome originally, if you were to look at how much DNA was contained, it would look kind of like that. And then during S, what's going to happen is at this point the cell has decided that it is going to at some point divide, so it's going to need an extra copy of the DNA, and so this is where it would now make an extra side, an extra copy. So it synthesizes a second chromatid so that when this cell divides later on, each new cell will get a full copy of the DNA. So again, if a cell was never going to divide again, it would never need to go to synthesis. It would just go into what's called G0, and it would just stay in that phase permanently. After S, it's called G2. There's certain proteins that are made in preparation for cell division. But again, this is another time when the cell is sort of doing all the jobs that a cell would normally do. M stands for mitosis. Technically, mitosis is really just a division of the nucleus. We think of it as asexual reproduction, asexual cell division, but if you really wanted to get technical, mitosis is actually just the division of the nucleus. It is possible that a cell could undergo mitosis, but that the rest of the cell doesn't divide. For example, your muscle cells do that. Your muscle cells undergo mitosis, and so they make extra copies of the nuclei, but then the rest of the cell doesn't divide. So you end up with one long fiber that's got, say, 100 nuclei on it because the whole cell didn't divide, just the nucleus did. So your cell is multi-nucleated, multiple nuclei. But generally, when cells go through mitosis, there's a separate event that happens at the same time near the end called cytokinesis. And cytokinesis means the division of the cytoplasm. So typically during late mitosis, anaphase to telophase, you'll see that the cell cytoplasm will actually divide also, so that you'll now end up with 2 cells and each cell has its own nucleus. But again, cytokinesis doesn't necessarily have to happen. It's not a phase of mitosis. It's a separate event that generally in normal cells, other than a few, like muscle cells, generally follows or goes along with mitosis at the end. Here's a little diagram just showing sort of how much time the cell spends in each of the phases. So if you see interphase, that's this gigantic section here, G1, S, and G2, and then mitosis is just this little portion, and cytokinesis generally happening at the end of that. Okay, So mitosis is made of stages, and you actually have a lab that you're supposed to do online where you're going to draw these and go through them, so I'm not going to spend a huge amount of time. This slide is really just showing you that it's a continuous process, and that if you really wanted to get technical, rather than just PMAT, prophase, metaphase, anaphase, telophase, you could actually go into, for example, pro-metaphase, which is the time in between prophase and metaphase, because the whole thing is sort of a continuous process. But we're just going to look at the 4 phases real quick. So prophase, you have your nucleolus, it disappears. The chromosomes condense, that means they coil up so you can actually see them. They now look like those X shapes. And you have this area called the centrosome, and that's where the microtubules are going to come from. In other words, if this is the cell, and here's our nucleus, it's going to start to disappear. And then you have these centrioles in animal cells. In plant cells, you actually don't have these centrioles, but you do have the area called the centrosome. And so there's our centrioles, and they're back basically moving to opposite sides, and then coming from those are going to be the microtubules. They're going to look like this. And in the next phase, they'll end up spreading all the way across the cell. So this is our microtubules, and then in here our chromosomes would now be condensing, coiling up so that you could actually see them a little better. They're probably going to look more like a mess when you look at a real cell. They're going to look like this. You're not actually going to see individual X's, but you're not going to see that gray mass anymore, the chromatin. You're actually going to see that there's these white gaps in the middle because they are actually coiled up. So that's the main thing that's happening here. And alright, so the centrosome replicates during interphase. That's the area where the centriole in animal cells is located. In plant cells, it's just an area. There's no actual little T-shaped centriole there. As a matter of fact, they've done some experiments where they've removed the centrioles and cell division still happened normally. So even though it says that it definitely is involved in organizing, they're not completely sure how important it is or what the role is of the centrioles. So they're going to move to opposite poles. That's what it's actually called, the poles. The middle is going to be called the equator. So just like you think of Earth having the North and South Pole and then the equator, the 2 poles are going to be on the opposite sides, and the center is going to be called the equator. And again, the spindle fibers are going to form, which are going to look like these little asters, these little stars coming out of each side. The nuclear envelope breaks down, meaning that the nucleus, you're not going to see it anymore. This little sketch on the right is supposed to be showing the centrioles moving to opposite sides and the chromosomes sort of coiling up. Alright, metaphase, really easy to see. Metaphase, the chromosomes line up down the middle, down the equator, and they're going to attach, the kinetochores, the proteins, are going to attach to the spindle fibers, and when they do that, that's going to allow them in the next phase to get pulled apart. So this is our, this is metaphase. This is a real cell in metaphase. You can see the spindle fibers here attached, and these are the chromosomes in the middle. When we draw it, we draw it a lot neater, but here's another diagram of it. It's 3-dimensional, so when we simplify it and sort of draw the little X shapes in a row, you have to think of it as more being a sphere. So this is metaphase. The next one's anaphase. At this point, the centromeres are going to split. The chromatids are now going to be chromosomes, individual chromosomes, and they're going to get pulled towards opposite sides of the cell. So the microtubules are going to shorten, and they're going to pull the chromosomes along with them to opposite sides. There's actually some of the spindle fibers that are all the way across the cell, and they actually get longer, so they're sort of pushing the cells apart at the same time that the microtubules attached to the chromosomes are shortening. Bottom line, it pulls them apart. And then lastly, you've got telophase and cytokinesis, and that's when basically the chromosomes are going to uncoil again. You're going to get 2 new nuclei. Cytokinesis, the remainder of the cell is going to break apart. It's different in plants versus animals. In animal cells, you get what's called cleavage. Actin and myosin microfilaments are involved in contracting and pinching the cell apart. But in plant cells, it's different. You get something called a cell plate that sort of forms down the middle, and that becomes a new cell wall. I have a diagram of it here. Here's cleavage on the left side in an animal cell. And in the right, there's a new cell plate forming in a plant cell. Alright, and real quick, this is an animation of it. This is a real cell in the little picture, and a sketch of what's happening. And as you see, there's metaphase, anaphase, telophase, and 2 new cells. And I'll play it one more time if we have enough time before it ends up.